Welcome back. We are starting the chapter 27 and we will talk about the microbial interaction in this chapter. So the first, we are looking at the many types of microbial interactions, and we're going to see, compare, contrast these different types of interactions. So the first thing that what we are going to talk about is the symbiosis, and this is an association of two or more different species of organisms. Now, many times we are going to see that these could be beneficial for both. One may be affected, the other may be not affected. Sometimes one will cause harm to the other one, and all sorts of relationship. Not only there are two, that many times we will see that there are more than two, even four or five different components or organisms are involved in this process. So here are just a couple of terms that we are going to discuss. Is ectosymbiont are the organism located on the surface of another, usually a larger organism. And this, you can just think about that some bacteria that are living on our skin, like Staphylococcus aureus or Staphylococcus epidermidis, these are very common. There are several different types that live on our skin. We can think about that these are our ectosymbiont. On the other hand, the endosymbiont is the organism located within another organism. And mainly the important thing that I want you to understand here, most of the time the endosymbionts are within the cell of another organism. That's the important part, within the cell. It is just not inside the animal, inside one of the cells. That is the most important part. So what is the relationship of the symbiont? Symbiont is any organism that has a specific relationship with another that can be characterized as a mutualism, cooperation, commensalism, but there are other amensalism, predation, also parasitism, and etc. So here we are looking at some of these different types of relationship, and we are starting with mutualism. So mutualism is a type of symbiosis in which the both partners actually gain from the association, and they are metabolically dependent on each other. So this relationship is obligatory. Both of them, A needs B, B needs A, and both of them benefit from each other. This is mutualism. The second one is the cooperation, where you have the A is positive but not obligatory interaction between the two organisms. And just the difference is that A is benefited from B, B is benefited from A, just similar to mutualism. But the difference is that mutualism is an obligatory relationship. You have to have that for the existence of both the different organisms. They will be benefited with the help of the other, but this is not obligatory. The next one is the commensalism, where the A helps B and B is benefited, but B does not do any positive or negative impact on A. So this relationship is just one-way traffic. The B, the second organism, doesn't do any harm, nor it benefits the A organism. On the other hand, the amensalism is a relationship which the product of one organism has a negative effect on the other organism. The organism itself doesn't really harm directly the B. It is a scenario where the product of the A will harm the B organism. Then we have other type of relationship like a predation. This is a relationship which one organism captures and feeds on the other. This is very straightforward. Then we have parasitism. Parasitism is a type of relationship where one organism captures and feeds on another one. And in this relationship, the one of the host which hosts the parasite is always harmed. So this is always a harmful relationship with the host. And the parasite is always benefited. Then we have this competition, and this competition is an interaction between the two organisms attempted to use the same resource, which is nutrient or space. And one may outcompete the other, or both of them can reduce their amount of population while sharing that resource. So it could be both. They're in one relationship, it can outcompete the other one. So the other has to find some other food source, for an example. On the other hand, the same food source is being shared between the A organism and the B organism, but both of them will grow in a reduced number. So these are the main different kinds of symbiosis that we are going to examine in this chapter. So first, takes a look at the mutualism. We're going to talk about these first. So here we are looking at the mutualism where some reciprocal benefit of both the partners. And this relationship with some degree of obligation, it's a mutualistic and the host are dependent on each other. And often the partners cannot live separately. That means that they are obligatory. So what are the different types of mutualism? There are many, many examples of that. And even we are not talking about even the human and microbial relationship. It is very, very complex. Let's take up some of the examples that we have studied in the laboratory and very well characterized. So sometimes these endosymbiotic microbes provide the needed vitamin and the amino acid in general to many of these insects. So we are talking about these two different insects, and we're going to give these examples. So one is the aphid and this Buchnera aphidicola interaction. We will come back to that in the next slide. But here we want to talk about this interesting relationship, which is an insect and Wolbachia relationship. So the Wolbachia, full name is Wolbachia pipentis. It's a rickettsial bacteria. So what is a rickettsia? A rickettsia is a very interesting group of bacteria. They are almost like viruses and they cannot live by themselves freely like other bacteria. What it means that they need a living cell or tissue to live in, or living cell in a tissue, just not tissue space, living cell to live. So they are completely obligatory and are endosymbiont as they live inside the cell. So they infect a large variety, this particular Wolbachia species, infect a large variety, more than 2 million insect species. They live in the cytoplasm of infected females. And very interestingly, they can modify fertilization event in the host. This is widely studied in a particular wasp called a Nasonia vitripennis. This is a particular wasp that is infected with this Wolbachia. If an infected male sperm try to fertilize an egg of an uninfected female, Wolbachia modifies the chromosome distribution as if the eggs were never fertilized. So they will fail to grow. But on the other hand, if an infected male sperm fertilizes an infected egg, both of them, male, female, both are infected with Wolbachia, the development is normal with normal sex distribution. So in other insects, Wolbachia simply kill all the male offsprings and the females develop just by cloning themselves, a process you know very well. We call this as a parthenogenesis. So this limits the genetic diversity, but this ensures that Wolbachia transmission is 100% among these. And this is a great success for this Wolbachia. Now, another interesting thing is that the scientists now trying to see that the Wolbachia actually also stop the virus from being spread by the viral vectors. And once that study has been done, now the scientists are trying to do something with the Aedes aegypti mosquito. This Aedes aegypti mosquito, you know that it's a very well biological vector for, say, Zika, dengue, West Nile, chikungunya virus. Many of these viruses are spread by this mosquito, Aedes aegypti. So the scientists are trying to infect these Aedes aegypti mosquito in the laboratory artificially, because the Aedes aegypti is not infected by Wolbachia normally. So it's a trans infection, it's an artificial infection. And the scientists have observed that, in fact, it is true that if these mosquitoes are infected with the Wolbachia, they will not be able to transmit the viruses. So Wolbachia blocks the viral transmission. So the scientists are working on this, that maybe at some point of time that they will be releasing these infected Aedes aegypti mosquito with the Wolbachia, and they can naturally mate with the wild types and then can reduce the amount of this mosquito which will be capable of carrying these viruses. So this is basically a biological vector control that can be done with the Wolbachia. So let's talk about the next interaction. So here, what we are looking at, that the mutualistic association is very common between the microbes of the insect, partly because the insect often consumes the plant sap or animal fluids lacking essential vitamins and amino acids. And these are provided by the bacteria symbionts in exchange for a safe habitat. So the first one we are looking at is a mature aphid. A mature aphid contains these Buchnera aphidicola bacteria within the specialized cell called bacteriocytes. The genome of these bacteria are very, very small, very interestingly, and we'll come back to that. But let us discuss that what they do. So these aphids feed on the plant sap. Unfortunately, the plant saps do not contain any of the nitrogenous material. That's the reason that they need a nitrogenous compound to synthesize their proteins. So these Buchnera aphidicola fix the nitrogen and produce the nitrogenous compound to these. On the other hand, the B. aphidicola is a safe haven for the food as well as shelter in these aphids. And it is interesting that the genome of these bacteria, they are very small, about 0.65 megabase. And this aphid and the Buchnera aphidicola relationship can be traced back to almost like 150 million years. So these aphids and these Buchnera aphidicola co-evolved over the period of time. And what has been shown that why their genome is so small, because they offer the very extreme genetic stability where there is no duplication of the genome, no translocation, inversion, or horizontal transfer. So why it is because as this relationship is extremely stable, this Buchnera aphidicola never tried to venture outside these aphids to look for anything. If they have tried to look for some other habitats, then there is need for some more genes which are needed for the survival of that bacteria. As these bacteria never tried to venture out to find something else, they need very little amount of gene for their survival because their environment never changed over the period of time. A classic example of coevolution. On the other hand, there is also a classic relationship in between a protozoa, termite, as well as many different type of bacterial termite relationship. So the termite provides a food for the protozoan. Protozoan digests the cellulose, and wood particles provide nutrients to the termite. And in this process, there are several different types of microbes involved. So the main thing that is important is that the termite feed on the wood, which contains the cellulose and the hemicellulose. And these are like 15, 20,000 glucose chains, and it is extremely difficult for the termite to break them down. Termite do themselves produce the cellulose enzyme to digest the cellulose, but that is not enough to break down these cellulose. So what they do, that they do have this digestion problem, and this is one. Another thing is that from the wood source, they do not get sufficient amount of nitrogen that is required there for their protein synthesis or the nucleic acid synthesis. So for this, they have a very complex symbiotic system in termites. So what you are seeing that right here, this is a picture of the Trichonympha species. This is a multiflagellated protozoan, and this protozoa here in purple lives in the termite gut. And along with that, there are several different type of bacteria which are also outside the Trichonympha in the termite gut. And there is also a type of microbe inside, which is known as elucimicrobium, that live inside that protozoa. So within the termite gut, the bacteria convert the nitrogen to the ammonia, which is taken up and converted to glutamine by the Trichonympha species. Along with that, they also digest the cellulose. The protist relies on this elucimicrobium to convert this glutamine to other amino acids, which are released with other cofactors that the protist cannot synthesize. Both the protist and the Trichonympha and elucimicrobium species also ferment glucose to acetate, hydrogen, and carbon dioxide. These products are exported by the protist and made available to the termite, which also relies on a free-swimming gut bacteria to boost the acetate level through the reductive acetyl-CoA pathway, and these are called the acetogens. In addition, the termite also harbors this methanogenic bacteria that will take up this carbon dioxide that is being produced and the hydrogen and convert them to methane, which will be excreted out. And it does not even stop there. There is also another bacteria, which are spirochetes. They also live on the outside of this Trichonympha protozoa. What they do, that these have the motility, and this motility keeps this Trichonympha inside the gut, because remember that the gut is motile, so the digested material is constantly being expelled through the gut. That gut motility may expel these Trichonympha, but these spirochetes, these are the undulating movement, corkscrew-like movement that will keep the Trichonympha inside the gut without being expelled. These are also very interesting, that how many different bacteria as well as a protozoa and the termite are in a symbiotic relationship. So the next example that we are looking at, mutualistic relationship, is the zooxanthellae. These are the marine invertebrates. They harbor these zooxanthellae, and these are dinoflagellate, and they provide this organic carbon to the host. And essentially, there's a species called Symbiodinium. This is a dinoflagellate, which provide almost 95% of the carbon they fix, because these are pigmented. And the coral has a pigment that protect the algae from the UV radiation. And also the coral provide the nitrogenous compound, phosphates, and carbon dioxide to these endosymbionts, because they feed on other chitinous animals as such, nitrogenous material provided to the zooxanthellae by these corals. On the other hand, what happens is a very interesting thing. Coral bleaching is the loss of either the photosynthetic pigments from the zooxanthellae or complete expulsion of these dinoflagellates, which are the zooxanthellae. So why this happens? This is caused by the temperature increase, and even a 2 degrees Celsius rise in the sea temperature can cause the corals to bleach out. And this has been seen in many parts of the world, and this is a concern. Coral bleaching will pose a danger to these ecosystems. So let's take a look at how this works. So here what we are looking at, that this is a typical rose coral, and this is green in color due to the abundant zooxanthellae within the tissue. And the corals provide this extensive habitat for a bacterial growth as well as the zooxanthellae growth. And here what we are seeing, that this is the bleaching of the coral. See that this part is bleached out, and that's what we are seeing. So let's take a look at the cross section of a coral and what they have. So outside, they do have a layer, which is a mucus layer. And this mucus layer contains different types of bacteria, one type of population. The bacteria in this mucus layer fix the nitrogen, because they also do this chitin degradation. So right here with the mouth, living part of the coral, this is called the gastrodermal cavity. And these corals, along with the water, get their food, and this could be many different types of tiny small crustaceans, and they do have an exoskeleton which is made up of chitin, and these bacteria can digest that chitin and provide the nitrogen. And here you can see that the zooxanthellae is lining the gastrodermal cavity. So as they are inside the gastrodermal cavity, the advantage is that they are protected from the ultraviolet, and they, using the nitrogen as well as carbon dioxide, which is provided by the coral, they thrive and fix the carbon dioxide into the sugars, which is utilized by the coral. Then there is a second group of bacteria which lives in the gastrodermal cavity as well, shown as these dots, and the exact function for them is not very well known. There is also another group which lives below this calicoblastic layer. And what is a calicoblastic epithelial cells? These are the epithelial cells which secrete and builds the exoskeleton, or the tough exoskeleton of the coral that we are familiar with. Inside this exoskeleton, or the coral skeleton, there are another third group of bacteria live in there. It is thought that these are actually very important for the nutrient recycling. So you can see that 3 different bacterial population as well as zooxanthellae all live in harmony in a symbiotic relationship, which is a mutualism. Another type of mutualism is a sulfide-based mutualism, and we are going to look at a tube worm example, which, along with the bacteria, which exists thousands of meters below the ocean surface. So we are going to see that these tube worms do have a relationship with these chemolithotropic bacterial endosymbionts, which lives inside the cells of these tube worms, and they are within a specialized organ called trophosome. So these bacteria live within the trophosome, and they help carbon dioxide fixation with the electrons provided by the hydrogen sulfide. So we are going to take a look at that. Before that, what we are looking at that in this figure summarizes the chemical reaction between the seawater and the rocks that occur varied range of temperature on the seafloor, the carbon and energy that support a diverse collection of microbial communities in specific niches within the vent system. So these are the hydrothermal vents through which these hot gases do pass out into the ocean floor, very deep ocean floor. The water don't boil at that temperature because of the pressure. The temperature could be very high. So let's take a look at what are these different niches. The first thing that what we are looking at is a very cold seawater, say from here. These are away from the vent, cold seawater, 2 degrees Celsius at that time. So they do contain oxygen, sulfates, and the carbon dioxide, and these seeps through the seafloor crack, and this is one of the community lives in there. Then there is another zone right here, which is shown as the mesophilic zone, and this is moderate temperature, about 15 degrees Celsius to about 45 degrees Celsius. This is high to about low oxygen, and microbes obtain energy by aerobic chemical reaction using hydrogen sulfide, methane, and hydrogen in the fluids, or metal sulfides in the rocks. These are some of the examples. Then we are going into a zone which we call as a thermophilic zone, high temperature, about 50 to 75 degrees Celsius in the thermophilic zone, and low to almost no oxygen. And the thermophilic microbes obtain their energy from anaerobic chemical reactions using hydrogen, sulfates, iron, organic carbon. These are some of the nutrients that is available to them. Then what we have is a hyperthermophilic zone, very high temperature, which is 80 degree to 125 degrees Celsius. And in the hyperthermophilic zone, this brown area, there is no oxygen, but there is still life in there. Hyperthermophilic microbes obtain energy from the end products of high temperature chemical reactions. And below that, which is the life does not exist. And the temperature here is about 130 to 140 degrees Celsius. And hydrogen, methane, carbon dioxide, and perhaps organic carbon created by the geothermal process by the microbes percolate upward via the cracks of the ocean crust from this area. So this is basically an overall biogeochemical cycle and the vent and related geological activity, what happens in different tiers, which is basically the cold and then mesophilic zone, then thermophilic and hyperthermophilic zone in the vent system. Now we are going to look at an example of this. So here you are seeing a community of tube worms, which is the Riftia species. So these are found in the Galapagos Rift, and they are in a depth of almost 2,500 meters depth, and they are near some hydrothermal vents. So here is a schematic of the worm, which could be a meter long, 3 feet long, more than 3 feet long. So they have these large gill plumes, which could be about 20 centimeter. And then they do have this particular area, which is known as this vestimentum. The vestimentum contains the brain and it contains the animal's heart. And the top end is the respiratory gill plume. And inside the trunk, the worm is a trophosome. And this trophosome consists of primarily these endosymbiotic bacteria inside the associated cells and the blood vessels. So the posterior end is known as the opistosome, which anchors these tube worms to the substrate. So on the other hand, here we are looking at that what are the different types of reactions that is going on in these tube worms. So what we are looking at, that just from the surrounding seawater using the gill plumes, you have oxygen, carbon dioxide, and hydrogen sulfide is taken in to this area. And the hydrogen sulfide can combine with the hemoglobin of this worm, and as well as carbon dioxide and oxygen can also combine to some extent. And this is provided to the bacteria in the trophosome. So once the bacteria gets this sulfide with the help of an oxygen, it produces the sulfur oxidation, produce this ATP and NADPH. These are the energy molecules. By the help of the ATP and NADPH, these bacteria fix carbon dioxide with the Calvin-Benson cycle. So these reduced carbon compounds, which are utilized by this worm, and this is their main carbon source. So these products are a little bit percolates into the worm, and these are used in the animal tissues. So on the other hand, these endosymbionts are having a safe harbor inside the gut of this animal. This is a very interesting type of endosymbiotic relationship and a mutualism at the very depth of the ocean where it is completely dark. There is no light in there, and still this mutualism exists. Very successful organism 2,500 feet below the ocean. The rumen ecosystem is very common among the ruminants, animals that have a stomach that is divided into 4 compartments, and they chew a cud. Why they have developed this process? Because this is basically eat first, digest later kind of evolution. Why they develop that? Sometimes when they were grazing, many times that due to the predation, their eating will be interrupted. So they will just run away and then digest later part. This is the reason how this relationship evolved. So if we just looking at the rumen is the upper part of the stomach, and this contains a large diverse population of microbes. So after the animal ingest the material, which is very rich in cellulose, they go into the rumen and they are liquefied, they are mixed with the saliva, and then the animal, when he finds time, basically chew the cud. So the material from here is regurgitated into the mouth. They chew it again, mixes with saliva, and it goes down into the rumen once again. And when they're liquefied enough with the action of the other bacteria, they also go through this reticulum, the second part. And from the reticulum, they will move into the omasum, which is a third part of their digestive tract. From the omasum, the material is going to move into the abomasum. This is the true stomach. And from there, they will move through the small intestine. And we're going to take a look in detail that what kind of a relationship happens here. So let's first take a look at an overview of that process before we go into the detail. So the ruminant microbial community have a mutualistic relationship, of course, and there are specific interaction within the microbial community happens. And in cows, the carbon dioxide, hydrogen, and acetate are used by the methanogenic archaea to generate the methane. But before that, there are lots of different things happen, and we're going to talk about that. So on the other hand, the very end, the methanogens synthesize some of these vitamins needed for their animal host. And also we're going to see that why they are very important for particular reactions, because they efficiently remove that hydrogen that is being produced by some of the previous reactions. If they don't, some of the reaction will not work. So let's take a look at that. So the first thing what happens that when the animal chews the material, it breaks down the cellulose. So one group of bacteria, they break down the cellulose and they just break these beta-1,4 linkages and they release the glucose. So this glucose is then converted into the acetate, then propionate and butyrate. And butyrate, propionate, acetate, these are the organic acids. And they also produce these fatty acids. And these are the true carbon source for the animal. And some of the ruminants, when they just produce the fatty acid, that's pretty much the end of their rumination. They do not undergo any further processing. But in the cows or the cattle, the processing is farther even once they produce the fatty acid and the organic acid. So what is seen in those, that there will be another group of bacteria, which we call as an acetogens. They are going to oxidize this and they're going to produce this carbon dioxide and hydrogen. Now, this process is also observed in somewhere else, which are in the flooded soil, where these acetogens live in there as well. So why we are trying to bring that example? Because both of these are kind of a similar relationship. There is a process which we call a syntrophy that is observed in the ruminants like a cattle, that is also observed into this flooded soil, where these acetogens produce this carbon dioxide and hydrogen and acetate by the oxidizing ethanol, organic acid, or other fermentation product. Now, the main problem is that these reactions are thermodynamically not favorable, because what happens that from when you try to produce this carbon dioxide, hydrogen, and acetate from these reactions have a positive delta G value. So as there is a positive delta G value, the forward reaction it is not favorable. You cannot just drive that reaction, but somehow these reactions are done. The enzymology is very complicated and not well understood as well, but it can be done due to some of these methanogenic bacteria like a methanospirillum, which lives along with what they do, that immediately when this carbon dioxide and hydrogen is produced, they convert this to the methane. That due to that, this hydrogen level is kept at very low levels. When they keep that hydrogen at this very low level, the previous reactions, which is the oxidation of the organic compounds, these reactions can be driven and the overall reaction happens. Because the carbon dioxide and hydrogen to methane, this is a negative delta G value for that reaction exists, and that reaction will go always towards the forward direction. But that reaction is needed to drive the previous reaction, which is the oxidation of the organic compounds. Only possible due to a low H2 level that is produced in that reaction, and that is helped by this methanogens. So there's an interesting example that one propionate oxidizing bacteria, it's known as Pelatomaculum thermopropinicum, and this uses a flagella not for the motility, but what they do that they use a flagellar protein which is known as FID, and this serves to basically as a transcription factor for the methanogenic or methanogenesis related transcription of genes in a Methanobacter thermoautotrophicus. This is a methanogenic bacteria. So you can see that there are lots of these reactions that happen. The acetogens do need to oxidize these organic compounds, the methanogens to live or drive these reactions. So this is a very nice symbiotic relationship, mutualism that exists not only inside the ruminants like cattle-like ruminants, but also outside in this flooded soil that we have talked about. The next type of symbiosis that we are going to talk about is a cooperation. And in this benefits both the organism in a relationship, but it is not obligatory. And this differs from the mutualism in this way. So here we are going to look at the interaction of a nematode, which is known as the Steinerna carpocapsae. So the Steinerna carpocapsae is a relationship with a bacteria which is known as the Xenorhabdus nematophilia. So this is the nematode right here, and this is a juvenile, and inside that you have this green bacteria that is shown in this figure, which is the Xenorhabdus nematophilia. Now here inside the gut, this bacteria lives inside the nematode gut and not expelled because this juvenile is not eating. So this nematode infects usually some of the caterpillars of the butterfly family in their developmental stage. And as the nematode feeds, what happens that this Xenorhabdus nematophilia is released and they produce toxins that will kill the host. But here is the interesting relationship. This bacteria produces an antimicrobial compound that prevent other microorganisms from consuming the insect cadaver. So this is a safe haven for the growth of the nematode as well as the bacteria. Now what you are seeing that the nematode matures and the Xenorhabdus nematophilia releases some of the maturing compounds which help the maturation of the nematodes as well as Once they are matured, they will mate and they will have all these juveniles. Once these juveniles come out, all these Xenorhabdus nematophilia will again infect them and live in their gut. This is a very interesting cooperative relationship where the bacteria helps a safe haven for the nematode to reproduce, and the bacteria finds a safe haven inside the gut of a nematode. So after that, we are looking at a process which is the commensalism. Here the one organism benefits and the other is neither harmed nor helped. So in the commensal, the spatial proximity of two partners permit the commensal to feed on the substance captured or ingested by the host, or the host commensal often obtains shelter by living either or in the host, and also can involve modification of the environment by one organism making more suited for another organism. So let's take a look at how we look at commensalism. So the microbial success during the spoilage of milk. See here, the fermenting bacteria promote the growth of acid-tolerant species. There are two ways that when these fermenting bacteria do fermentation, they are going to produce an environment which is rich in organic acid. And these organic acid will allow the growth of acid-tolerant species. This also is to some extent true that how these fermenting species help in producing, say, buttermilk or yogurt production. So in the yogurt production, these fermenting bacteria promote these acid-tolerant species, which are beneficial bacteria. Here it is not as. Also, the formation of a biofilm, the initial colonizer helps other microorganisms to attach that. So for an example, we talked about these biofilms, if it's in a tooth plaque. So the initial organism produce that slimy layer on top of the coated tooth with the protein, and then other organisms come in and they attach to that, and this is also another way that the commensalism work. On the other hand, the amensalism is a negative impact of one organism or another based on release of a specific product. We are going to see some example of antibiotic production by the fungi and how the bacteria can be killed. So a classic example of that, we are going to take a look at the diagram in the next slide. There are some ants which are known as these atine ants. These atine ants, cultivate a garden of fungi belonging to the genus Leucoprinus. And this is dependent upon an actinobacteria in the genus Pseudonocardia that produce inhibitory compounds that prevents the growth of this Escopsis species. This is a predatory fungus for the Leucoprinus. This will destroy that fungal garden. So this is a nice way that the ants do carry this Pseudonocardia on their body so that they cannot destroy this Leucoprinus species. So let's take a look at this relationship. So these atine ants cultivate their own nourishment, a garden of fungi belonging to this genus Leucoprinus. And their ability to accomplish this advanced task depends upon this actinobacteria right here, actinobacteria in the genus of Pseudonocardia. This bacteria produce an inhibitory compound right here, which stops the growth of this Escopsis species, which is detrimental for this Leucoprinus. This is a parasite for this fungal garden. So the ant developed a morphology to carry this actinomycetes, this Pseudonocardia, on their legs and the body. On the other hand, along with this relationship 1, 2, 3, and 4, there is a fifth member which is also involved in here. This is an actinomycetes yeast type of fungus, which is known as the Phyalophora. So this Phyalophora is a predatory fungus which feeds on these Pseudonocardia. So what is the advantage of that? So this allows, this predation prevents the cheaters. So what we mean by that, when the ant carries these actinomycetes could be many different ones, and the production of these antimicrobial controls, which they will produce here as well, antimicrobial controls is energetically very demanding. So many of these bacteria will not produce these antimicrobial all the time. whereas some of their neighbors will. So this Phallophora species only will feed onto the pseudonocardia which are not producing this antimicrobial, because if they're producing the antimicrobial, they will not be able to, because this is a fungus as well of actinomycetes. So if the pseudonocardia is producing antimicrobials, this will not be able to feed on those. Here, think about that in this complex of the pseudonocardia, you have a mix of population. Some of these bacteria are producing the antimicrobial agents. Some of the bacteria are just not producing the antimicrobial agents. It is just the choice of the bacteria to be energetically conserving the energy versus producing that. So this Phallophora species will only be able to eat the microbes or the pseudonocardia which is not producing the antimicrobial agents, because that will kill the Phallophora as well. So the only ones which are producing this antimicrobial are going to thrive here. So that means the cheaters, means the ones which are not producing the antimicrobial, are eliminated from this population. A very interesting relationship. So after that, we are looking at another, these are very straightforward, the predation. So among the microbes involves the predator species that attacks, usually kills its prey, and at the microbiological level, this is also present. This Vampirococcus has an epibiotic mode of attacking the prey, and there are two different types. So for example, this Daptobacter penetrates the prey and directly consume on the cytoplasmic content, whereas this Delovibrio penetrates the cell wall, grows outside the plasma membrane. This is essentially in the area which is the periplasmic space. Then there is a Myxococcus. This use the gliding motility to creep, overtake their prey, and release degradative enzymes. And these benefits include the increased rate of nutrient cycling, but these are predation is also very common at the bacterial level. So here are some of the examples that we just saw in the previous slide. So here what it shows right here is a Vampirococcus right here attached to the outer membrane, and they secrete these degradative enzymes that will result in lysis and release of the prey's cytoplasmic content. In here, we are seeing a Daptobacter which can penetrate the cell, and once they're inside the cell, they will consume the cytoplasmic content as well. So here in this picture, you are seeing the Myxococcus xanthus and E. coli colony. Initially, you have these two drops, and then gradually what happens that this Myxococcus moves over and they consume the E. coli, and this colony becomes clear. And you can see that they swarm over the prey, and this bacteria has a characteristic by which they moves in waves. So the parasitism is one organism, the parasite gains, whereas the other is harmed, which is the host. And always some coexistence between the host and the parasite happens. And the successful parasites have evolved to coexist in equilibrium with their host, because if the balance is upset, the host or parasite may die. So this is a very interesting relationship. So here we are looking at an example of parasitism. And in this here, you are looking at a photograph of a flattened leaf-shaped genus, Umbilicaria. And this is a common foliose lichen, whereas here you are looking at a thin slice of this under a compound microscope. And this is the green algae occur at the thin upper layer. And these are the fungal hyphae that make up the rest of the lichen. So what is the type of relationship? So before going to the controlled parasitism, let's take a look at that. What are the partners and what are the definitions? So in here, the mycobiont is a fungal partner, which provides water, minerals, sheltered environment, and firm substratum for growth of the lichens. Whereas the phycobiont or the algae or the cyanobacterial partner do have the pigments, photosynthetic pigments, which provides the organic carbon and the oxygen. So if you think about this relationship, these looks more like a mutualistic relationship compared to a parasitism. And why this is a parasitism? The reason that we call this as a controlled parasitism, because if we independently grow these phycobiont, which is an algae or the cyanobacteria, so without this mycobiont, this phycobiont, which is the algae or the cyanobacteria, will grow much faster. So what happens that in a mutualistic relationship, the growth of this microbiont and the phycobiont both should be dependent on each other. So that was the mutualistic relationship. So this is not mutualistic. Why this is not a cooperation? So in the cooperation, what will happen that if suppose this algae or cyanobacteria is growing, and in the absence of the fungus or the phycobiont, this algae or the cyanobacteria will grow slower. But that's not the case. What happens if you do not have the phycobiont, this will grow faster. So basically the microbiome is the parasite of the phycobiont. So a relationship which looks like more of a mutualistic relationship is actually a controlled parasitism in the lichen. So what is the outcome of this long-term parasitic relationship? What happens, the parasite loses this unused genomic information because they do survive only inside their host cell. And a classic relationship is the aphid endosymbiont, the Buchnero aphidicola. We saw that. There are many human pathogens also, which is Mycobacterium lepori causes leprosy, Mycoplasma genitalia. Then you have this Encephalotoxin cuniculi. And these are some of the human pathogens which do have a very reduced amount of genome due to their parasitic nature. The last one that we are looking at is a competition occur when two organisms try to use the same resource. And there are two possible outcomes of the competition, as we talked before, that one organism may dominate, and this competitive exclusion principle, where two organisms share the resource, both survive at a lower population level. But in one case, one organism may dominate and may completely exclude the other one or eliminate the other one, where both can live in harmony to a level where they both survive, but at a lower population level. So these are some of the overview of the relationship that the microbes do have, along with other microbes, as well as the eukaryotes as well. These are the microbial relationships. And they rarely lived alone. They evolved the specific ways with other organisms, which includes the microbes, plants, and animals as well. So that will be the end of this chapter.